So I'm going to talk to you today about genetic approaches to dystonia in general and also to spasmodic dysphonia in particular. This is just a slide to tell us about classification of dystonia, genetic forms of dystonia, and so you can see down here on the bottom there's about 20 different forms of dystonia, what we call loci, and about 12 of these now have genes identified for them. So in parentheses are the genes that have been identified. And the other thing that I want to tell you about this classification is that it's really a mixture of all different kinds of disorders. And so up here we have primary dystonias, dystonia plus, and paroxysmal dyskinesias. And I'm going to focus, because focal dystonias fall into primary dystonias, I'm going to focus on primary dystonias. And I'm going to tell you a little bit about THAP1, and then we'll go into focal dystonias, for which we have no real genes for yet. So before I start, I want to talk a little bit about inheritance. And so dominant inheritance, this is a family tree, or what we call a pedigree. The squares represent males, the circles represent females. People who are colored in are affected with a disease. And so when we talk about dominant inheritance for any particular trait, we have two copies, one that comes from our mom and one that comes from our dad. And so when you have a dominant disease, you only need to have one bad copy. You don't have to have two. So if you have two bad copies, that's called recessive. If you had one bad copy, that's called dominant. And so what I want you to see here is that most people who are affected have a parent who's affected. All right? So you only need one bad copy, and most people who are affected have an affected parent. So I'm going to now talk about DYT6 dystonia, which is an early onset form of dystonia, and it was identified first in a very large family. There's 23 affected members. This is a pedigree that's connected 6 generations back, and it's from the Amish Mennonite population. And it's an early onset form, as I said. The average age is about 16 years in this family. It's inherited as a dominant trait. And it's mainly, the onset is mainly in the arm, but there's other body regions involved, with speech often affected, which is why we're interested in it in relation to spasmodic dysphonia. And so this gene was identified by my lab in 2009, and it's a gene called THAP1. And so from 2009 until now, people have searched families all over the world, and this shows you all the different mutations that have been identified in this gene. So there's lots of different mutations of this gene identified in lots of different families. And so we have done a study where we looked at 544 patients. 43 of them were generalized, with generalized dystonia. 17 had multifocal, so they had a much earlier age of onset, about 19. Then we had 129 segmental dystonias and 355 focal dystonias with a later onset, age of onset of about 39. And you can see these numbers in parentheses. 7% of the generalized had mutations in THAP, 1.5% of the segmentals, but less than 1% of the focals. And so there's another study done by Zhao et al. In 2010 that also had mainly focal dystonias. And so when we look between our study and their study, there were 500, almost 600 people who only had sporadic spasmodic dysphonia. And so of those about 600 people, only 4 of them had mutations in this gene. So that's less than 1%. It's about 0.7%. And so this clearly tells us that you can have a mutation in this gene that causes your spasmodic dysphonia, but it's a very rare event. And so this tells us that, in fact, spasmodic dysphonia can be genetic. And so if we look at the clinical features of all of the different people who have had mutations identified, what's the typical clinical picture that we see here? Typical clinical picture is an early age of onset, so about 13 years, but you can see this wide range. So people who are as old as onset of age 62 had mutation in this gene. This main site of onset is in the arm, about 50% in the arm. More than 50% have generalized or multifocal dystonia, but 10% of people do remain focal. And the thing that's important here, of course, is that speech involvement is quite prominent. So among these folks that have generalized dystonia or multifocal dystonia, one of the body regions that's involved is speech, which is certainly not typical for the other early onset gene, DYT1 dystonia. And so depending on the clinical features, the phenotype, THAP1 mutations account for about 1% in adult onset, but can be as high as 25% in early onset non-focal cases of dystonia. So this again tells us that there can be a genetic mutation that by itself causes spasmodic dysphonia. So the other thing that the other concept that I want to get across, the other genetic concept I want to talk to you about is something called reduced penetrance. So this is the other gene, DYT1 dystonia, for early onset dystonia. And these are some pedigrees. And this is supposed to also be an autosomal dominant disorder. And we saw the original pedigree, and I told you that everybody who's affected usually has an affected parent. So reduced penetrance is when a person has the mutation but does not show the clinical signs. All right, so for instance, again, people who are colored in are affected. People who have dots actually are carriers because we know what this gene is. We can assess all the family members. So if you look at this pedigree, we have 2 people who are second cousins, but nobody in between is actually affected, yet they're all gene carriers. So that's what reduced penetrance is. So in DYT1 form of dystonia, the penetrance is about 30%, which means you see a lot of people who don't have affected parents, and yet it's still genetic. So why is that important for focal dystonias? So focal dystonias, adult onset focal dystonias, are actually the majority of primary dystonias, about 90%. But we do think that genetics is involved because although many cases appear singleton, between 5 to 25% have an affected relative. So within spasmodic dysphonia cases, about 12% of people who have spasmodic dysphonia have a relative who is also affected with some form of dystonia. It doesn't have to be spasmodic dysphonia. It can be torticollis, it can be blepharospasm, it can be writer's cramp, but some other form of dystonia. And so that suggests again that there's some genetics, some genes involved here somehow. But what makes it hard to study this is that most of these focal dystonias are late onset. So if we want to get DNA from a whole family, a lot of times parents are already dead because the person who's affected was affected when they were 50 or 60. And when we want to look at their children are too young to be at the age of onset, so we don't know if they're not affected yet, and we don't know if they're going to be affected. So we really don't have these kind of pedigrees that we had to find the DYT6 gene, where we had 23 affected people all in the same family. The other thing that complicates it is when we look at the folks who have other relatives, their pattern of inheritance is actually consistent with autosomal dominant disorder, but with reduced penetrance. Now I showed you those DYT1 pedigrees, they had 30% penetrance. We're thinking adult onset focal dystonia is 15% or less. So that means there's lots of people who don't express the trait, but who may carry a gene. The other alternative is that, and these aren't mutually exclusive, is that it's multifactorial inheritance, which means that there's multiple genes that interact with each other and that may also interact with some environmental insults that we don't really know what those would be. So now there's been a revolution in human genetics, and it started with the Human Genome Project, and probably took us about 10 years to sequence one person's genome. It probably took us about $2 or more billion to sequence that one person's genome. And now, so a genome, everybody's genome consists of 3 billion bases. So we have 3 billion pieces in our genome. And now we can probably sequence a person's genome in about 5 days and for about between $5,000 and $10,000. So a huge improvement over the Human Genome Project. And that cost is coming down. And so in the next 2 to 5 years, we'll probably be able to get all of our genome sequenced for less than $1,000 if we want to do that. But right now, the cost prohibits sequencing our whole genome. But the thing that's interesting about our genome is that the genes, the coding parts of our genome, the parts that make proteins, it's only about 2% of our genome. And most often, the mutations that cause disease are located in the genes, in these protein coding parts. So if we could pull out just this 2% and sequence that, that would be much more cost effective and more bang for our buck because we could find our mutations, hopefully. And so there's a strategy where we can actually do that, where we take DNA and we break it up randomly. So we use our scissors and we cut it up. And then we have what we call baits that are to each of the genes in our genome. And so just like we're fishing, we use our baits for the genes for the genome, and we pull out all the genes. And then we take them and we put them on our big machine that costs, you know, $500,000 or $1 million, and we sequence. And so this is what it looks like. So this is a representation of a gene where the horizontal lines are what we call introns, and the vertical lines are what we call exons. So these are actually the coding bits. The vertical lines are the coding bits. And so what we're doing here is called exome sequencing. We're sequencing all of the exons from all of the genes in a person's genome. And so this is just showing you when we go fishing, we actually get what we want. So this represents that we have captured this little bit that represents the exon. And here you can see that these peaks are higher every time we have an exon, and we're not really capturing very much in this region, which is a part that we really didn't want. But you have to be aware that sometimes we don't capture everything. So we probably capture about 90% of the exons in our genome. So if our mutation of interest is in this exon, we're not going to find it because we never captured it. So we have to be aware of that. So this is the strategy that we use. We basically take our cases and we exome sequence them, and we find a bunch of variants. So these things are called single nucleotide polymorphisms, or SNPs. We have a bunch of variants, and they're hopefully in the genes. And it turns out that there's a database, or lots of different databases, of SNPs that are found in the normal population. So because dystonia is rare, the chances that a person is in the database that has dystonia is quite rare. And so we look at all the variations that are already known in the database, and we exclude them from being causative for our disease of interest. And so then we've come up with our variants that are unique to our cases, and we ask what genes are those in. And so this is just sort of saying the same thing in words, that exome sequencing is very valuable for these rare Mendelian traits. Regardless, we don't need to have a family with 23 affecteds. We can have a family with 1 or 2 or 3 affecteds. So any family becomes valuable now for exome sequencing. So we're going to filter out all the common variants by looking at what's in the databases. And then we're going to look for only the things that are shared among the affected family members, because presumably, if they have a gene in common, a defect in common, they're going to have to share that. And then we're going to look for if that variation that we find that's in common in the people that we sequence, if it also segregates in the family. If there's other affected people, do they have it? If they're unaffected people, do they not have it? With, of course, the caveat that we have this penetrance issue. So there's going to be some people who are unaffected that still have it. But we know that that's going to happen. And so then we're going to, if we find a mutation, then we're going to look in other families with a similar phenotype or even sporadic patients and ask if this gene is important in those families. And so these are just 2 early onset families that we started with, and we took 4 affected members from these families, and we sequenced them, and you can see each person has about between 200 and 250,000 unique, variants within their genome. And so if we look at what these 4 people all share, we're down to 60,000 variants. If we do some quality control, we're down to 4,000 or 5,000 variants. If we now take away all the things that are in the known databases, we're down to 200 variants. And then if we look at where these occur in the gene and are they functional, we're down to 20 variants. So we've come from 200, 250,000 variants down to 20 variants that could potentially be one of them, the thing that's causing the disease in this family. So that's the kind of thing that we're interested in doing. And I am happy to tell you that we just received this month, April, end of March, a grant from the Dystonia Medical Research Foundation to try to identify a spasmodic dysphonia gene using this technique. And a talented instructor in my lab, Tanya Fuchs, wrote this grant and has received this grant. And I was actually told, I don't know if Kim can confirm this, but I was actually told last week from Dystonia Medical Research Foundation that part of the funding for this grant is going to come from the NSDA. Right, and so, that's your guys' hard work coming to fruition and helping us. And so we're very thankful for that. And so this is the family, which doesn't look good. I went from a Mac to a PC. So this family you can't see. This family basically has 3 affected first cousins, and one of those first cousins has a affected child. And so there's 4 affected members in the family, and we're going to exome sequence these guys probably the end of May, beginning of June. And so then what we'll follow up on is if we can find a variant that is only in this family and in an interesting gene, then we're going to look in other SD families in the same gene and also in SD sporadic cases and ask if this gene is important in those other populations. So another concept I want to move to is something called an endophenotype. So this is a subclinical marker of gene expression. So what does that mean? So there's a particular endophenotype I want to talk about. It's called the temporal discrimination threshold test. And this was developed by a group in Ireland. And basically, this is a test where you have 2 stimuli that are applied to, say, your finger. And it turns out that when you have, so the average person can distinguish the 2 stimuli at about 20 milliseconds. But a person with different kinds of adult onset dystonia takes about 40 milliseconds to distinguish those 2 stimuli. So it doesn't make you go to the doctor. You don't really notice it in your life, right? So it's this subclinical phenotype, but it turns out that all different forms, so torticollis, writer's cramp, spasmodic dysphonia, blepharospasm, and even musicians have this difference in this temporal discrimination test. So it looks like it's potentially a marker for the disease. So if you then look at what we call first-degree relatives, so first-degree relatives are your parents, your siblings, and your children. So if you look at first-degree relatives, it turns out, so these are people who aren't necessarily affected. You can see that in these first-degree relatives, they also, some proportion of them, show this abnormality. So if this is a marker for carrying a gene related to adult onset dystonia, we can actually use this trait instead of the affected status. So here's a pedigree, and the people who are black, colored in, are affected with this type of dystonia, with an adult onset dystonia, but all these guys who have red circles have an abnormal TDT. So that expands the amount of people we can look at in a family from just these 5 people to all these other guys. So we can do genetics on the TDT, this subphenotype, this endophenotype, as opposed to just trying to find these families, which are actually quite rare to find, with multiple affected members with adult onset dystonia. And so this just shows, again, now they're looking at 32 patients with sporadic adult onset. So these aren't people who know that there's other family members who have dystonia. These are people who think they're the only person in their family who has dystonia. And if you look again at the siblings and the offspring, a certain proportion of them have abnormal TDT, suggesting again that this looks genetic, even in people who don't seem to have other affected family members. And so we can use this to try to find genes, to expand our pedigrees, to get more people, to do this exome sequencing. And so, of course, what we have to do first is, for instance, try to find the gene in this family using the affected folks, and then when we find the gene, look to see if all of these people who have abnormal TDTs actually have the mutation. That way we can validate that this is a valid test of carriage of a gene for dystonia. The last thing I want to talk about is another study that we just got funded, and this is led by Christine Simeon at Mount Sinai, who's an imager, and this is imaging genetics of spasmodic dysphonia, and this was funded by NIH. And so this is another study specifically for spasmodic dysphonia, and the purpose of this study is to identify changes in the brain in patients with spasmodic dysphonia and correlate them with genetic risk factors. And so this is sort of, again, looking for these sort of endophenotypes to ask if imaging can act as an endophenotype. And so the idea, we just heard that perhaps what we call SD is maybe a bunch of different diseases. It's really not just SD. And so we may be able to, using this, try to improve detection and diagnosis and use for alternative treatment options when treatments become available, because we may be able to subcategorize folks either by their imaging pattern or what genes they have involved or things like that, and they may respond differently to different treatments, for instance. So this is just, I have to say right up front, I don't know much about imaging, so this is from Christina. So this is just an example of different changes that are seen in SD patients versus controls. So what we want to look at are, are these changes present in all forms of SD? So we want to look at both AD and AB forms of SD. And we also want to look at people who have a family history of dystonia and people who don't have a family history of dystonia, and ask, are there differences based on these simple clinical criteria that we can distinguish people with? And the differences may not actually be related to the categories we're going to make. And so then after that, we would take a certain proportion of these people and we would exome sequence them, and we would look for variants that correlate with the different patterns that we're finding. So here we're not looking for a single change like we are in the families. Here we're looking for a group of changes that then define a certain pattern. So eventually you could actually, instead of imaging people, you could take those variants and do them on people and say, well, you should be this type or you should be that type. So we're going to correlate the variations with the imaging. And so this just goes through the inclusion and the exclusion criteria. And this is the procedure that's going to happen. You'd be, once you agreed to participate, you would undergo a telephone screening to try to decide if you meet the criteria, inclusion criteria. You'd go through a detailed clinical examination. You'd get blood drawn or saliva. We can get DNA from both saliva and blood. And then you would come and you'd get imaged. You'd get an MRI. And so this is just the advertisement, which I'm pretty sure is going to be on the website. And then this is the information about how you can, I don't know if you have the sign up here?
We will have it out later today.
Okay, so later today they'll have a sign up sheet where you can sign up, or you can take a survey on SurveyMonkey to see if you're eligible, or you can email us or call us if you're interested in participating. And so I'd like to finish up with, why are we even trying to find genes? So finding new genes may help us to identify or connect functional pathways, which will then provide us with therapeutic targets. And as I said before, it may also help us define SD subtypes that can be important for directing therapy. All right, and of course I want to thank our funding sources and all of you, because we certainly couldn't do any of our studies without your participation. So thank you very much.